Good morning, everyone. And I welcome you all to the Foundation Day of National Institute of Immunology. Today, the 6th of October, holds a special significance for us at the National Institute of Immunology. It's a day of honor, reflecting on our past achievements while also looking ahead to the future. I am Banya, and it's my great privilege to welcome you all to the 38th Foundation Day of the National Institute of Immunology. Let us celebrate the remarkable journey we have had and the exciting possibilities that lie ahead of us. I extend the warmest wishes from the NII family as I welcome our esteemed guest of honor, Professor Rahul Purwar, faculty at IIT Bombay and founder of ImmunoAct, India's first gene-modified cell therapy company. We are truly honored to have with us today our founding director, Professor G.P. Talwar. Welcome, sir. I also warmly welcome our director, Dr. Debashish Mohanty. At this moment, may I invite Dr. Mohanty to present a token of our heartfelt appreciation to Professor G.P. Talwar. Thank you, Dr. Mohanty. I also request you to please present a token of our appreciation to Professor Purwar. I would like to extend a warm welcome to all the dignitaries, especially the dignitaries from the DBT family, scientists, researchers, NII alumni, staff and students who have joined us today to celebrate and cherish the spirit of NII. Thank you for being here. Elie Metchnikoff, recognized as a father of innate immunity, eloquently described the immune response of a body as a complex interplay between many different factors, a symphony of cells working in harmony, illuminating the intricate dynamics of the immune system while highlighting how various cells and processes collaborate and work together to safeguard the body. This metaphor of a symphony beautifully illustrates how each component plays a vital role. And this concept of symphony and harmony resonates profoundly with the NII family today. As a research organization, we have grown from strength to strength because every individual and every effort has contributed to our collective success. Together, we embody the harmony that drives our mission forward. Since its inception in 1981 and formal establishment in 1986, the Institute has been at the forefront of unraveling the intricacies of the immune system. We engage in cutting-edge research across diverse fields, including immunology, infectious diseases, reproductive biology, structural, chemical biology, genetics, cell signaling, and cancer, all under this very roof. Today, we stand as a proud organization committed to addressing global health challenges with a clear vision for the future. This decade-long journey has been made possible by a rock-solid foundation built on vision, knowledge, experience, excellence, and leadership. Today is not just a day of celebration, but also a moment of memory and reflection. It's a time for us to come together, look forward, and ask ourselves, where do we go from here? And for that, I would like to invite and request our leader, the director of NII, Dr. Debashish Mohanty, to deliver the welcome address.
Sure. Namaskar, good morning. Professor Rahul Purwar, our chief guest for today's event, Professor Talwar, our founder director, all the guests and dignitaries, and Dr. Alka Sharma, senior advisor DBT, and the directors of the other autonomous institutes, and my faculty colleagues at NII, our students, staff of NII, our alumni, former colleagues. I welcome you all to the 38th Foundation Day ceremony of National Institute of Immunology. I thank you all for your enthusiastic participation in this event this year, even on a Sunday. So this represents the spirit of NII. So every year we have been celebrating the Foundation Day of NII on 6th October to commemorate the day on which NII was dedicated to the nation by the then Prime Minister in 1986. And however, this NII society, as Banya said, has been established in 1981, and it involved several years of hard work to build this campus. And we are very happy to note that, like every other year, Professor Talwar and his family members have joined us for the celebration of the Foundation Day, and he just celebrated his 98th birthday yesterday, and even after that, next morning he is here because NII is something very close to his heart, so he can never miss NII. We are of course missing Mrs. Talwar here. She is slightly unwell, but she has conveyed her wishes to you all. Yesterday I met them in their birthday celebration. So, coming to NII was established, as I said, in 81 when government identified basic and applied immunology as a priority area in recognition of its potential impact on human health. In the first decade, NII between up to 91-92, NII made major contributions in the area of vaccine development, reproductive immunology, and most notable being the successful development of leprosy vaccine using Mycobacterium indicus pranae. So it may be important to mention that utility of MIP as an immune modulator has led to exploration its efficacy for sepsis and also cancer immunotherapy in recent years. So in the next decade, during 91 to next 2 decades, 91 to 2011, so the NII further strengthened its position as a premier research institute in India and made innovative contributions in basic immunology, infectious disease research, virology, chemical biology, structural and computational biology, as evident from several high-impact publications. So during the last 13 years, NII has acquired cutting-edge modern infrastructure for research and has made notable contributions in cancer biology, systems immunology, aging research, glycobiology, structure-based drug discovery. NII has also initiated new research programs in areas like virus immunology, single-cell immunogenomics, nanotechnology-based therapeutics, microbiome, and organokines. So during the period, NII also strengthened its PhD program, and currently we take students twice a year in both in the July as well as the January session, and we attract 25 to 30 bright and talented researchers for PhD to our PhD program through a highly competitive examination. We held examination almost in 30 centers all over the country, which is also followed by an interview. So that speaks the attraction NII has among the young researchers. So apart from our publication and patents, the success of NII is also evident from the fact that 8 of its faculty members have been recognized by Shanti Swarup Bhatnagar Prize, which is one of the prestigious awards in India, and also the NII faculties have significantly high share in the fellowship of the academies, and 10 of our faculty members have gone to take up leadership positions in other research institutes, and that includes Dr. Rajesh Gokhale, the current secretary of the Department of Biotechnology. So many of our alumni are holding senior positions in academics and industry within the country and abroad. So today is also the occasion to remember the contribution of all our former directors, starting from Professor Talwar to all the former directors as well as our former faculties. And it is needless to mention that success of NII is not only because of our faculties and research scholars, but also it is our extremely dedicated technical, administrative, and support staff. I also thank Secretary and senior officers of DBT for their generous support to NII in all our endeavors. And now, it is time for us to consolidate our efforts to get ourselves ready for future challenges. Using NII, NII was started in 1981, it was even before the DBT started, and using NII as a role model, DBT had set up 14 other autonomous institutes covering different areas like cell science, plant and biomedical genomics, in areas like neuroscience, food and agribiotic, animal biotech, stem cell, bioresources, etc. So government has envisaged that all these institutes should work together to play an effective role in development of circular bioeconomy. And in November 2023, just a little about a year back, DBT has set up BRIC, Biotech Research Innovation Council, as an umbrella organization under which these 13 AIs will operate with their own functional autonomy, and network programs can be executed in a much easier fashion. That is why now we are BRIC NII, and this obviously this cannot escape the attention of Professor Talwar, and the first question he asked me is what is this BRIC? So BRIC is just an umbrella organization under which we can all work together. This one. So now, in addition to that, own autonomy, this one, and now onwards, we BRIC institutes where students will will also work together. In last July, we started a joint PhD program between 15 BRIC institutes where students will work in interinstitutional collaborative project, and we have selected around 75 students for all 15 institutes. This is a new collaborative research program which has started. And this very recently, and in August, government announced the Bio-E3 policy, that is biotechnology for economy, environment, and employment. And size of bioeconomy is currently $130 billion, and it is likely to reach $300 billion by 2030. And BRIC NII will be expected to play a bigger role not only in vaccine research and disease biology, but also in development of therapeutic antibody, new vaccine platforms, precision biotherapeutics. So it will be necessary for us to integrate laboratory research with translational health science and public health, which was the original mandate with which Professor Talwar had started this institute. And so in recognition of this challenge, NII had initiated several flagship, integrative programs, in like a flagship program on human immunology, a network program with clinical centers for genome sequencing of drug-resistant tuberculosis, collaborative research program with European Union for development of universal flu vaccine, and also a collaborative program with another DBT institute in Northeast, IBSD, for deciphering molecular basis of many traditional medicines. So these collaborative research programs have started yielding interesting result. So I have to thank my colleague, Dr. Pushkar Sharma, for his initiative. Many of these collaborative programs started when he was the acting director, and he still takes active interest in pursuing them. And of course, needless to say, all our faculties who are participating in this program, so I must thank them. So in addition, 4 new interinstitutional collaborative programs are being planned under the new this BRIC PhD program. One of the mandate of this program, every PhD student will join a project. There should be at least 3 principal investigators. They should come from 2 different institutes. So under this program, we have 4 different programs covering in areas like cancer biology and studying vaccine efficacy and neuroscience, and also relating to another program relating to crosstalk between dengue and diabetes and things like that. So these are really interesting programs, and students have been attracted. So hence I am confident NII can play a leading role in DBT's autonomous ambitious program in the area of biomanufacturing under this Bio-E3 policy for promoting circular bioeconomy. And normally in the Foundation Day, we don't present slides, but Professor Talwar always asks me, you tell me what was the achievement of NII this last 1 year. So I would just quickly go over the work of NII during, so we have been working in several disease-related areas: infection immunity, human immunology, tuberculosis, malaria, cancer biology, neurodegenerative disease, aging, and metabolic disorders. And now this is under BRIC. All these 14 institutes are going to work together. And some of the recent achievements has been in the area of in the immunity and infection, like immune cell clues for development of treatment of inflammatory bowel disease, and single cell RNA-seq for CMV and identifying CMV specific T cells. These are newer latest technique using which you can identify new cell types, and evidence again for development of multi-organ strategies to treat IBD, and the role of SGBP and in anti-SCV activity. And under human immunology, we have a T cell assay platform which I mentioned last year, and this is the human immunology program where Dr. Bina and Dr. Nimes are involved. That is what we, and a few other, Dr. Anis and some other faculties are involved. And of course, NIA has been active in malaria research from Dr. Puskar's group. New pathways have been identified which could potentially give drug targets. Dr. Agam's work has a very interesting studies. He got an Indian patent for a new malaria vaccine. And in cancer biology, Dr. Sagar Sen Gupta, though he has moved as director of NIBMG, his research group is still active here, and they have identified small molecules for adjunct therapy in colon cancer. And Dr. Sanjeev Das group had made very interesting studies, leads in the area of cancer biology, and also, of course, the anti-cancer effect of gut metabolite indexyl sulfate has been identified by Dr. Anil Kumar's group. And this is what the network program on sequencing of drug-resistant tuberculosis, and Dr. Binay's group has identified role of drug resistance and links to the novel DNA repair pathways. And of course, on tuberculosis, a new drug target and a new scaffold, and on which a US patent has been filed, and Dr. Biswal's group has been very active in this area. Coming to neurodegenerative disorder, Dr. Sarika's group has done Triphala, improves the cognitive function in murine models of Alzheimer's disease, and of course the role of microRNA have been identified for Alzheimer's disease in the Dr. Puskar's group, and a new the clomipyramine as a new molecule for treatment for Alzheimer's disease, for which a US patent has been granted, and a biomarker using a gut-derived metabolite. This also has been identified from Dr. Anil's group. And we are also active in the area of aging and metabolic disorder, and there are interesting studies. In the interest of time, I will skip there. And of course, not only we should do our research, we have to be in the outreach program. So this is Dr. Banya, who is our public relation and science communication expert. She has been very active to attract students, and we have organized several programs. And this is the one has been just set up in Dholpur, and this is government's program of in every aspirational district. These institutes have to set up a science center or a museum, and thanks to Dr. Basist, our head of administration, he has been this has been just set up few days back. It would be inaugurated. This is a new thing in the Dholpur. So with this, now I won't take much time, and we always, of course, look forward to the Foundation Day talk, which is very special to all of us. Today's Foundation Day talk is by Dr. Rahul Purwar, who is a professor in the Department of Bioscience and Bioengineering at IIT Mumbai, and he is one of the youngest professor who has been extremely successful, both as a extremely talented researcher, also as an entrepreneur. He is the founder of Immunology, a successful biotech venture. He is well known not only for completely indigenous development of India's first CAR T cell therapy, but also making it available to the patients through his company Immunology at a fraction of the cost in international market. His vision to democratize access to advanced cell and gene therapies in India, I am sure many more interesting things are going to come from his group, and he will provide us valuable insights into how to integrate laboratory research with translational health science. And I must thank Rahul Purwar for accepting our invitation and agreeing to give this talk at a very short notice. And finally, looking forward to a memorable Founders Day celebration. I thank you all.
Thank you, Dr. Mohanty, for the address. I now welcome Zikra, our young researcher in her 5th year of doctoral studies focused on designing a malaria vaccine, to introduce Professor Rahul Purwar to the audience.
Thank you, Dr. Banya. It's my pleasure to introduce the speaker to the audience. Professor Rahul Purwar is a pioneer in cancer immunotherapy and a distinguished academic professor of biosciences and bioengineering at IIT Bombay. He holds a PhD in molecular medicine from Hanover Medical School, Germany, and an MSc in biochemistry from the University of Lucknow, India. Professor Rahul's professional journey spans various prestigious institutions and roles. He started his postdoctoral career with a fellowship at Hanover Medical School, Germany, and then moved to Harvard Medical School, USA, as a research fellow. His industry experience includes a stint as a research scientist at Immunology Incorporated, Waltham, Massachusetts. Since 2022, he has also been serving as the founder of ImmunoAct in Mumbai. In this role, he drives strategic initiatives through accelerated process and clinical development and manufacturing, which has not only led to the successful market authorization of the country's first CAR T cell therapy, but also one that is fully indigenous to India and distinguished by a superior safety profile. This is the first of many planned milestones in democratizing access to advanced cell and gene therapies in India, his fundamental vision and founding principle of ImmunoAct. Professor Purwar's research work is well recognized in the scientific community with numerous publications to his credit in esteemed journals. His contributions to the field of biosciences and bioengineering are commendable, and his work continues to inspire many in the scientific community. With a huge round of applause, please join me in welcoming Professor Rahul Purwar to the stage.
Good morning, everyone, and thank you, Jikra, for a kind introduction. And a huge thanks to Dr. Mohanty and entire NII team for having me here. I'm really delighted to be here, and it's really an honor to speak about some of our work in front of the pioneers like Dr. Talwar, Dr. Alka Sharma, Dr. Mohanty, Dr. Sagar, Dr. Sinha, and many other dignitaries and students. I know that I think what I will do in the Founders Day, many of you know more immunology than myself, so I'm not going to show you too much data. But what I will do is I will show our journey, how we have done what we have done in last 11 years almost, being a faculty member at IIT Bombay, and then how I started my entrepreneurial journey in 2018, and how the journey was in last like 4 or 5 years as an entrepreneur. Okay. So there's some issue. So this journey started almost like 11 years ago in IIT Bombay as a faculty member when I joined. An idea there was that in the West, we have these state-of-the-art technologies or the advanced therapies which are available there, but they reach India after several decades or sometimes even don't make it. One of such therapy is gene therapy. Gene therapy, some of you might have heard, it costs approximately somewhere between 5 crore to 20 crore rupees per patient for one injection. 5 crore to 20 crore rupees for per injection is a huge amount of the cost. Majority of us will be unable to afford these highly expensive therapies. And that's one of the reasons why these industry do not come to India and they don't find market here. And that's why these therapies are not available. Our vision was, can we make it? And then can we make them affordable? And that's how was the motivation we started. You all know that India is pharmacy of the world. We have proven it especially in even COVID again and again, right? In COVID, it was a really testimony. India, DBT efforts, ICMR efforts, they have been tremendous. We supplied vaccine to the entire world. We are very good in chemistry. I think we are probably number one in chemistry in small molecules, drugs, manufacturing of these by chemical synthesis. But we are getting there in biologics. And we were nowhere on the world map in emerging cell or emerging new therapies when it comes to cell therapies, cell and gene therapies, or gene therapies. But if you see that industry is moving towards the personalized, precise, and safe medicine, and emerging new therapies could be one of the modalities. And these modalities, these new therapies, they really provides the hope of these individuals. What you see in this girl is the 6-year-old kid got cancer, and these are the cancers which are not curable. They basically that no medicine, or when all medicines stopped working, and that's the time CAR T or these advanced therapies help them, and this kid is now alive and cancer free, at least until now. It's been more than 2 years, kid is still alive. I think hopefully she will be through, cancer will not come back, that's our hope. Gentleman again was a medical technician, had cancer, no hope, again given the hope using these therapies. And these examples and many more, whether it's a soccer player, national level cyclist, or medical technician, some student in the lab, some Bollywood, some farmer, dentist, and even this patient. And many of you know cancer patient, one of the problem becomes they cannot be eligible for, or they cannot make kids, but he fathered a baby very recently. So these are the examples that gives you thrill that whatever we have done in 10 years, it is literally making a difference in the life. And not, these are just few examples, and there are many more. And problem of cancer is huge. There are so many patients worldwide. In India, again, there are so many patients, and mortality is very high in our country. Although incidence of cancer, when you see, you may know that incidence is low. If you see a number of cancer patients per 100,000 people in our country is very low compared to West. But mortality is high because we don't have those medicines which can cure for long or which can be curative. Right? One of the major obstacles why these therapies are not available is mainly the cost. I think we have other things, but cost becomes a huge challenge. And here you see the world map. This is old world map where you see the gene therapy or cell therapy. They're all clustered either in this part of the world, or even if you come here, this side, mainly our neighbor. So if you just put a line, and so global north has it, global south almost nothing. India did not have anything. Africa almost empty. LATAM very little happening. But everything was dense towards the US, Canada, China, little bit Russia, some in Australia, right? And I think we have done it. We have done many, we have good stories about our mission, Mars mission, and many other missions. This is not just many places we have proven that India can be innovative. We are innovation, like we are culture where innovation happens, right? And we do innovation at the cost fraction of the cost. What Gravity movie, you know, the budget of this was much more than our entire Mars mission. So at affordable cost, we can do it. And this point I want to really highlight, and I want to give the take-home message also. Lower cost does not mean low quality. What India is generally perceived, and this is a perception, that when you reduce the cost, it is reduce the quality. And this is the example now I give. You have this Fairfield Inn in Marriott, same Marriott chain in New York, and same Marriott chain where I come from, Mumbai, right? Cost is almost same price, you get both places and you notice the difference. So what I want to say is that when we develop something at the lower cost, does not mean the lower quality. And in an innovative country, we can change that perception and we can develop good quality products at affordable cost, and that will really solve the world problem. It's same like when the TVs were LCD TV, they were 10 lakh cost. Today LCD TV cost is very little. Many examples, like I think until Narayan Murthy started the IT business, computer was for only elite class. Now computer, phone, they are from everybody's pocket and everybody's lap, right? So with this, how we have worked hard from 2013 when I joined as a faculty member at IIT Bombay. So in some time, like 4 or 5 years, we were only developing the platform, which is lentiviral vector platform. We moved any drug development, first you develop the product prototype. So this you consider as a lab research, as a product prototype. Then you come into translational mode when you do the validation in certain animal models. Then you go to the phase 1 of the human clinical trial. So phase 1 of the trial started somewhere in 20. Then 21 onwards, we started doing phase 2 clinical trial and then scale up. And the October 23, CDSCO approved the product for multi-market use. So that means now this product is available across the country at the many hospitals, like almost 75 plus hospital now in India literally use gene therapy. So gene therapy is no more a science fiction, it's actually a reality in our country now, right? Opportunity in this field is huge. So as a student, as another young faculty member, if you are thinking about research area, I think cell and gene therapy has huge potential. Because as we go further, you will realize that this therapy is not just the solution for 1 or 2 diseases. It is a platform which can be used from heart attack to diabetes, which are like a big issue right now in our country, along with cancer and autoimmune disorders, right? So if you see if you are an entrepreneur thinking about market, etc., this like $1 billion opportunities is there. This is going to be more and more. Now you will see that US FDA in next 5 to 10 years is going to approve at least 20 to 35 gene therapies in this world. So gene therapy will be the, I think, focus in next 5 to 10 years. That's why if you see the trends, the way US FDA approve it, there are so many companies, startup, even the large pharma, they are investing humongously in the cell and gene therapy portfolio, right? Right now, if you see US FDA or EMA, both together, they have cell and gene therapy or market approved these advanced therapies in hematological disorder. So blood related disorders like cancer, non-malignant disorder like sickle cell disease, thalassemia. Now imagine, for example, sickle cell disease is a huge problem of our country. We have high number of patients, but we don't have the technology. The part of the world where patient number is very little, they have the technology. So it's like need is some other part of the world, the technology is another part of the world. But how to bridge those gaps, I think that is what we should think, and if you are a student, you should think in this way, how we fill the gap of this need in our country. Because sickle cell disease, which you might have seen, government is also putting a lot of effort, right? Sickle cell disease, the gene therapy that came, it got 15 crore rupees per patient. Our sickle cell patients are mainly the tribals, and the tribal belt, like Gujarat and that area has huge number of sickle cell patients. Can you imagine that 15 crore per patient can we invest in them, or can they afford, even can government even help? It's very difficult. So unless we find our own solution, sickle cell is not going anywhere, right? So hematological malignancies, oncologic disorders, dermatology, skin disorders, neuro, opthal, musculoskeletal, like DMD you might have heard. There's a lot of focus there. Immunodeficiency, aesthetic as well. India has still only one cell and gene therapy is approved, which is Nexcar 19. And this, so what is this CAR T cell therapy, right? CAR T is, this is the revolution, I would say, done by Carl June from University of Pennsylvania. So he is the pioneer in the CAR T field, because he started the first animal experiment he did. The vector was developed somewhere else, but he has proven in the lab that CAR T really works. And then he took this CAR T to first 3 patients. One of them was Emily Whitehead, who you, if you type CAR T, then Emily Whitehead becomes the poster child for that CAR T work. Steve Rosenberg does a lot of work on the cellular therapies. So when Dr. Carl June, who is pioneer of this thing, when he visited India in 2020, and this slide actually I have prepared for him because he wanted like what are the challenges and how he can help us. So when he visited in 2020 in Hyderabad in BioAsia conference, he made a statement to the media that India's involvement can make treatment mainstream, says this CAR T pioneer. Unless, same thing, like if you again go back to analogy of the Narayan Murthy or those kind of that IT giants, they make really the IT, computer, and all this as a main thing in India. All of us use it, no matter you are elite, middle class, lower middle class, or even the poor. So once I think India comes in picture, we think about scale. So our population, our young people, our hardworking young professionals, I think they will make, or they will ensure that not only just CAR T, all cell gene therapy platforms, some of you working in immunology, NK cell, gamma delta T cell, B cell, macrophages, people are making CAR B, CAR macrophage, CAR NK, CAR gamma delta, not just CAR T. So this field has a lot of potential. What is CAR T? Just to give you a little idea. So CAR T is basically when once patient come into the clinic, the patient came to clinic, doctors take the blood from this patient and send it to the laboratory. In the lab, these T cells then genetically modify, and once they are genetically modified, they express a protein. Imagine this is a protein, just like other protein, chimeric antigen receptor. This protein is sitting on the T cell surface, and then once it is there, this cell becomes a cancer fighting cell. These CAR T are cancer fighting, so whenever they will find the cancer cell, they will get attached to them and try to kill. That's a very simple, in a layman language, very simple mechanism of CAR T. These cells you enhance in numbers in the lab by culture and then send them back to the patient. So these are autologous platform. That means the entire process you have to repeat for each patient. So unlike paracetamol or other antibiotics, other chemistry-based drug, you can go simply to the medical pharmacy and can get it. CAR T is not like that. CAR T is a personalized medicine. It's autologous. So for every patient, you have to make a batch. If you think about pharmacy or pharmaceutical industry language, every patient is for one patient, one drug, one batch. Did not want to go, because you are immunology student, you understand, that's why just showing you slide, that the CAR T anatomy looks like this, like any other receptor. Outer surface comes from single chain variable fragment, which recognizes cancer cell. Then there's a transmembrane domain that makes sure that it is sitting on the cell membrane. And the CD28, this entire is a co-stimulatory domain. Some CAR would have CD28 as co-stimulatory domain. Some would have 41BB. Some would have OX40 or TNF alpha family. Some will have only one. Some would have multiple. So depending on what type of synthetic biology you want to use. So it's all about your choice. As a scientist, you want to have what type of CAR T you need. You can design those CARs, and then they will fall either first generation, second generation, third generation, and so on. So now, not only we have CAR, we also call them trucks, because more and more biomolecules you start putting in your construct, the car start becoming larger. So it's no more become car, they call it truck or track. This is this science journal named CAR T as the most phenomenal advances in medical science in last 100 years. And probably the Emily Whitehead and her foundation is very active in this proof of concept study. There's a long-term cure. This word cure was never existing in the field of cancer because everybody would call it cancer under remission. You will never hear clinician even accepting that cancer can be cured. It's always when it is not there, it's remission. But here, after 10 years, I think they, UPenn and Carl June and Emily Whitehead Foundation, they are now advocating that it can be cured. Many CAR T are there in the field from $500,000 US, as I was saying, 4 to 5 crore rupees for the product alone, and then remaining 4 to 5 crore goes into the clinical care. So what cost to patient come almost $1 million. And how you reduce the $1 million, that was a question, right? This platform is not just for the blood cancer. Recently, the heart attack, as I was mentioning, cardiac fibrosis can be treated with engineered T cell. The many other cancer, including the solid tumor. Success is very little in solid tumor like breast cancer, glioblastoma. But as I was mentioning to someone in the morning, the benchmarking of the CAR T is now that everything should work more than 50% of the patient. But even if the patient like glioblastoma, which is a very uncurable or untreatable cancer, if even success is 10 to 20%, it could be a great success. But now first CAR T has created so much hype, so much expectation became so high, it's very hard to convince people that from $1 million you are only doing 20% of the benefit. So I think this war will happen, but I think we will settle somewhere. Okay. Recently, if some of you are following these articles like this research area, there's huge, many publications suddenly came in last few months in Lancet or Nature Medicine or many other journal, New England Journal, about lupus. Same CAR T which works in the leukemia and lymphoma that also works in the systemic lupus erythematosus, SLE. And this is showing, so you all know as an immunologist that autoimmune disorders are because of high autoantibodies. Autoantibodies are produced by plasma cells which comes from the B cell origin. If you are removing B cells by CD19 CAR T, you will have some success in curing or at least putting these difficult disease in check. And that's what exactly the concept was, and it works. We also, now India, when our economy is doing better, our infectious disease are on the decline. At the same time, you see our young generation has a lot of allergies and autoimmune disorders. And that's the place we will also start. So in India, we have a lot of SLE patients who are bedridden. They can't walk, they have a lot of problems. So now we are also starting the clinical trial with CD19 on this platform. So finally, this Nature wrote an news report on us that we have created this made in India CAR T at 1/10 of the cost. So now I will focus how we have our story, like when we wanted, how we improve the access of this CAR T. Our objective was, can we make it affordable, more than 10x, reduce the cost more than 90%, and then make at a scale. This, we did not want to have a boutique therapy, as I said, not for elite, but rather many patients, or at least as many patients who can get, who can benefit from it, they should get the CAR T cell therapy. That was the idea. And to ensuring that idea works, we make a team. So IIT Bombay, where I come from, the part of the product and technology development, Tata Hospital, Dr. Gaurav Narula and Dr. Hasmukh Jain, these were the leaders for the clinical trials. A knowledge partner, National Cancer Institute, NCI, NIH, who was the subject matter expertise. Dr. Terry Fry was there earlier, but now he's moved to a private company. But Dr. Nirali Shah is heading the pediatric oncology group there. So both of them, they helped in the clinical side. Dr. Steve Heifel, again from NCI, he helped us with the GMP and GMP related manufacturing processes, and Immunoact become a vehicle to take it to the masses. And this is what is very important. So I will come back again on this, that we all as a researcher, we are very comfortable here at this part. How to do the collaboration with the clinician, this is a hurdle, but it's a less barrier, but biggest barrier comes when you take it to the masses. So those are the things how we navigate it through, we will discuss a little bit. But the best way to navigate all these or make your own path through all these issues is collaboration and partnership. I think this is where we should promote a lot, that how we collaborate and bring your complementary thing, what you don't have. For example, we are very good in research, but we may not know a lot about regulatory. How you bring the regulatory support from a partner, or how you bring clinical support from a partner, from the clinical side, or then how you go and ahead and find the marketing people. And that's the place where you're investing in infrastructure, education, and training helps you. What we were doing is, as a company or even as a professor, what I had thought, I will never marry to the one particular idea. We want to be adaptable. Wherever technology takes us, we will move. That's why we created a platform which is very much adaptable. Many of you who works in this area would realize that there are some very simple solution. People will tell you there's a box. In that box, you can make the CAR T or make the gene therapy. We never got very excited about those ideas, probably because we wanted to create the platform. That platform which is adaptable, which is ultimately help these patients at an affordable cost. So whenever technology mature, when my LCD TV technology mature, I can continue adapting to that particular technology right at that time. So I am advancing as technology advances. I am not stuck to one technology platform. That's what I meant by platform, not a particular equipment or not particular technology. So it should be adaptable as per consumer needs. It should be scalable, not no more just few patients we can do. We should do scale at a scale that becomes disruptive. So anything in this world, if we want to do it, we have to do at scale. If we are small, nobody hears us. When you are in the group, everybody listens us. Same thing in the science. If we are at masses, do gene therapy and cell and gene therapy, if 100 scientists do it, I think Dr. Alka ma'am, DBT, everybody will listen to us. They will fund us, right, when we come together. So this is very important, that cell and gene therapy, we should do many more people, and then it creates a very good ecosystem in the country. Right? And this is how you lead the innovation, right? That's how world see you. If you are only one product company, one lab which works on cell and gene therapy, nobody cares. We have to, as a country, as multiple institute, even IIT Bombay should collaborate to NII, JNU, etc., to make that many technologies come. Right? This just one slide, entire 10-year research, because I don't want to bore you with the data, because data you see in all the presentation. What we have done, at least we have put India on this global map of gene therapy, which I showed you in the beginning. There was no dot in India. At least we have put a dot in the global map. We were unique. We published the work. We patented it. We tested it in very thoroughly in the laboratory, in the animal model, now in the trial also. And we were first in the clinic. And this work, if you are interested, this is our CAR. And I don't know, not many people read this work, but when NCI wrote this story again, so I think many people and many of the journal also took from NCI when they spoke about it, that India's first homegrown gene therapy has roots in NCI collaboration. So until this part, all of you do also very well. All of you are expert in doing your own research, publishing it, and making patent. What comes here is a bigger challenge, because that's not our comfort zone. And as I said, to cross that barrier, that how to take lab work to the market, I think that is the challenge. And that challenge we solve by creating good advisors, good team. Dr. Carl June, when I met him for the first time, almost now 7 or 8 years back, when I explained them we are doing this and how we are approaching it, 6 to 7 years back, he said, looks like you will make it. Finally, we made it, and he graciously said, we will help you. We will help you developing India's program. And now he is in our scientific advisory board. Dr. Ravindranath, I don't know, some of you know him, but he is the owner of Global Hospital or Gleneagles, which is one of the private chain in India. Dr. Satwa Nilapu, he is professor at MD Anderson. He ran the Gilead first CD19, CD28 clinical trial. So there are two pioneers of CAR-T. Both are here. No competition, not worried about anything. They are helping us. So he ran the Gilead trial, which is a second product. He ran the first Novartis trial, first product that comes to Novartis. Both gentlemen, when we explain them the data, they are willing to help. Message to the young star is that please ask for help, you will get it. If you ask the people to help you, I am 100% sure they all will be willing to help. Dr. Gaurav, you know, both of us work very well together. Avery and same department at Carl June. He works on GD2 cars. Somebody was telling me they work on GD2. So he has a lot of experience in GD2. Nirali is, again, the pioneer in bispecific car, again at NCI. Dr. Satyanarayan Chava, he is Laurus Lab founder and chairman. He invested in us. Dr. Hasmukh Jain, as I mentioned, he is the medical oncologist who ran the trial that lead to the market approval. And Steve, again, manufacturing side. And these folks, they are from the business. And Atharva was my student, PhD student. So PhD students, again, you should feel Atharva is a very young person when he joined me as a MSc student, converted his MSc into PhD, and now he's co-founder of the ImmunoAct as well, just like me. Taking a lab research to the market, he is now actually, even at very young, he is very, very young, and he lead entire scientific group at such a young age. So to make translation, this thing you have to create ecosystem where business people there, where data people there, where your own scientists are there, clinical side is there, business who can invest in you. So this kind of team, I think, is the key for the success if you want to take your lab research to the translational mode. Then, of course, you have to develop all these kind of state-of-the-art GMP facility. I have personal opinion, academic lab, this kind of GMP doesn't work very well because these are very hard to manage. So better you create in a place where it can be managed properly. You do your hard work in the lab and then let somebody who do these kind of work, managing facility is a pain. And continue keeping it as per regulatory compliances is really not your cup of tea. That somebody let these people who can do these kind of facility manage these things. So we created a huge facility around 30,000 square feet, 2 floors. One floor is just dedicated to GMP and another is office. And we invested a lot in the collaboration to make the translation. Alone we cannot do, but I think with the help of the experts across the world, across India, we really worked very, very hard. So develop the team, I think that's the message number one. And second is, this is another area where we are not good at. We are not expert. We can learn, but we are not expert of the regulatory. Learn, I think, regulatory by discussing with the stakeholders. In India, we have so many committees. I think all of us know it. But this is a place where we have to work with the government, with the different departments, discuss with them, learn from them. We will make mistakes. Nobody is expert. Neither they are expert who are evaluating your application, nor we are expert who are writing those applications. We have to work together, right? So that's what I do. When I don't know it, I make mistake. They teaches us something, we teaches them back, learn it. And that's the only way. So without cribbing, we have to move on in this. This is, I think, the lesson. Because we have to go for not just our institutional committees, we have to go to the state committees. From state, you have to come to central agencies. And everywhere, same thing will be discussed 3 different times. So it takes long, but finally you will make it. This is a really complex logistic. Another challenge you will face when you come to these are not, in academia you don't have these challenges. These are the challenges you feel when you come to take it your lab research to the translational part. Lot of logistics, which again we are not expert. How to ensure everything is happening nicely at the hospital. In hospital in our country, our clinicians don't have time. They're super busy in OPD. They have hundreds of patients waiting. You go to AIMS or you go Tata Hospital. They don't have time. How you ensure that all the paperwork is properly completed? So you might have heard these are clinical research organizations, CROs, etc. But you have to work together to ensure, especially cell and gene therapy, which you are doing for first time in the country, such a complex thing. Data is documented properly because this was the first clinical trial of the CGT. Our image is at stake, right? If we do anything wrong, everything falls through. So this was a very hard part. We learnt a lot in several years. Then logistics, making CAR-T in the lab. So this was our expertise. This was not a challenge that much because that's what we all do and we can do. This part again, how to ship at the room 4 degree and how ship back at the -80. Lot of, when as an academic person, you will feel just shipping at -80, what is the big deal, right? But when you go to those, all the SOPs which are there to just simply ship a product, it is a tremendous work. And that's the place where you learn something new. And then how to infuse. And of course, doctors are there to treat the patient. I don't think we have problem in the delivery. Our doctors are expert, our scientists are expert. Neither I don't see any problem. If any of you want to do this work, you can do it. Our doctors, don't underestimate, they can deliver the CAR-T. No problem. They can manage all the toxic. Our problems are these very simple. So, and you know, if you have done PhD, I had a privilege to have breakfast in the morning with some PhDs, very bright students. I think you all can do it. This was the moment of truth where the first CAR-T was given in 2021 to a pediatric patient. First child we have treated, and we have shown you the photo also of one of those kids. The one who treated unfortunately passed away. But later we have many kids who are still alive and cancer free. After that, we have treated patient by Dr. Hasmukh and Dr. Manju Sengar in the adult side, which is trial for 15-year older. And these were the moments when you feel that your lab research finally went to the clinic for the first time, and first time in India we had treated patient. Just to show you, there are many patients now, there are more than 12 months, because we are also young. Our data is only 12 months or 24 months old. So this is leukemia data, all these kids. You see these responses. Some of you, if you are working in the cancer field, this kind of response you don't see in any of the cancer medicine. 73% CR rate, complete response. That means cancer is completely gone. And that's the reason why there is a hype on CAR-T. Of course, some of them will relapse. So if you see this patient, he relapsed even after 7-8 months. He relapsed after 4 months, these two. This one also relapsed. But see some, they are still completely cancer free and keep going. And these are the motivation you get as a scientist. When I was PhD student, I used to say, if I save one life in my research, I will be taking retirement and nicely sitting on the couch and thinking, I have done my bit. And this is something really make me happy. It's like not only one life, I will show you many now. Many examples are there. And these patients, now you see the green bar, green line if you see, almost 80% of the patients, if you treat them right in time, they are cancer free for until 12 months. What the data is, cancer hasn't come back, they are cancer free completely. But you have to pick them at right time. And right time here is if you have a cancer which is less than 25% B cell, which are cancerous B cell in the peripheral blood, in bone marrow. If your bone marrow of the patient has less than 25% blast, you can really have very good result. But if you have too many blasts, disease has gone haywire, and then you decrease this 40%. And this is our entire population, which comes somewhere between 60 to 70%. So now, again, field is evolving. Lot of R&D is needed to find out who are these people. Can you find them out before? Or you are going in this line, in the middle, you take these as well as these, but these are expensive therapy. I think it's better to find biomarkers which will ensure which patient will get benefit. Similar story in the lymphoma. As some of you are clinicians, then you will know this disease is really bad disease here. Those who can read the PET scan, those who are clinicians, you will realize this is really bad disease. This is not that bad, but it's a big tumor. And now you see within 30 days of the treatment, disease is completely gone. Same here and same here. Right? And now you see the results here. Same thing. So these patients, these are high-grade lymphomas as well as low-grade lymphomas. You see there are patients who are surviving more than 24 months. And these are the patients who never had any hope, because they were told almost go to palliative care. So these are the patients who were in palliative care, and now the patient I told you, for example, this one even fathered a baby. It's a big thing, right? And then similar story in some of these. But again, two things you can learn from here is, of course, there's a relapse. Cancer comes back. Cancer is a devastating disease. Despite such an expensive treatment, despite having these good technologies, cancer will come back in many. Not only that, this unrelated death. And unrelated death means cancer did not come, but patient is still passed away. Probably or most likely because of infections. Because all these patients become immunosuppressed because of the B-cell aplasia, which the CD19 CAR-T does, and sometimes they also die. So we have long way to go. Lot of work is needed, how to prevent all of this, but this is a baby step. It is not the end. It is just like we are the toddler now. We have long way to go to improve all of these outcomes, how to save these people, or how to prevent relapse, how to make this graph from 24 months to take 10 years. Then only the technology is really proven. But just to give you a little idea how we are performing compared to the world class. Are we really world class? So these are the entire world CAR-T. Tisacell, Axicell, Breyanzi. They are for the lymphomas. So here is our CAR-T, NextCAR19. These were all results of phase 1, phase 2 trial only. So it's really all, not apple to apple, not 100% comparison, but at some level there is a comparison. So you can see our CAR-T is as good as all of these. So efficacy wise, overall response rate is very good. How we are doing in a leukemia cohort. So these are the leukemia studies from all these Tisacell and Brexel. These are the phase 1 and 2 studies for this particular indication. How we are doing here, I think we are doing very similar. Right? So we have shown that compared to all other CAR-T which are available across the world, we are doing in efficacy very similar. But how we are in the safety, is our CAR-T safe? This is the most interesting point. Again, this is early data, so don't completely take it with a pinch of salt, but at least early sign is promising. Of course, later we need more data, and this will tell the further truth. But as of now, what you see, just neurotoxicity. In our car, at least in the phase 1, phase 2 trial, we saw 0% neurotoxicity of the grade 3. And here you see other cars. And that's what we are saying, that if our neurotoxicity is low, our hospitalization will be low. And because our hospitalization is low, our clinical care cost will be low. And that will make it world's probably the most affordable car. Right? So if you see ICU admission, the patient after CAR-T, how many go to ICU? At our place, it's only 7% patient end up in the ICU. However, in other CAR-T, whether you say Zuma 7, don't worry about these words, but it is just showing that these are the different products and the different trial data, 24% to 47%, or half of these patients end up going to the ICU. At our place, only 7% patient going ICU because we have less CRS and less ICANS, which is neurotoxicity. Okay? So this is how you then calculate something called healthcare resource utilization. For a given drug, how much healthcare uses you use. In our place, is fairly less, and that's the reason we can reduce the cost burden or financial burden to the patient. So this was about clinical side. Now just giving you an idea about how are we achieving scale, because that was our major idea. Now you see that India, not only just in Mumbai, we are in Delhi, many centers in Delhi are giving CAR-T. Chhattisgarh, as far as Chhattisgarh, Bhubaneswar, and I think in East, Kolkata also have done CAR-T. Maharashtra is doing a lot, Gujarat, Andhra. Basically, in all part of the country, we are treating patient and CAR-T is available. If you see, just remove Tata Hospital, these are the major centers beyond Tata Hospital, they are also doing the CAR-T. Just giving you an idea that in the map, at least is getting much and much blue as we go along. And the model which we wanted is that regionally decentralized model. Right now we have only facility in Mumbai, but if the scale goes high, we can maybe set up another facility in Delhi or Bangalore, depending on the need. Right? So, but always CAR-T comes to these centers, centralized production of the vector, and we can have the quality assurance team in the backbone as a centralized facility. And we are investing really huge now in infrastructure. We are coming up with our own building where we will have all end-to-end production of raw material, not every raw material, mainly the plasmid, vector, CAR-T in this facility, so that we can put things on scale, so that many hospitals can be served. Then we are also now, this is right now Make in India effort. In our patient, it's not just in India had a vacuum, other part of the world like Africa, Latin America, they also don't have CAR-T. Mexico, for example, is Foreign Minister of Mexico, who visited us in Mumbai. He went to the entire lab. He was so excited. He said, we also have economy similar to India. Can we also have the CAR-T? So now we have going to start soon the clinical trial in Mexico for our product with the help of a couple of hospitals they have identified. Actually, he's the Director General of that hospital. And most likely we will start the trial either end of the year or early next year. So now this was about access. Now coming to the delivery of this most affordable CAR-T. As I mentioned, the delivery is, I think, is very difficult because if you see our healthcare affordability gap, very few patients have these insurances. Very few patient has employee scheme like CGHS, ESIS. Government schemes are again very few, and there is a big population, uncovered population. So everybody, like half of the people, now things are improving. Of course, from 86% to 7 in 21, we have 46%. So there's every time it is improving, and these schemes are definitely helping. All the government schemes, they're helping a lot, but there is gap. There is affordability gap. So if you see how many patients are there truly and how many can finally afford. So we have, even for this particular indication where NexCAR19 or CAR-T can help, we have 89,000 patients or 90,000 patients. Relapse and refractory, if you just say 40%, these patients can be benefited with the advanced therapies. But hardly these patients can afford, that's what we feel. And right now, I don't think even 5 to 8,000, because right now 500 patients only. So how we brought the cost, we are feeling very happy that we brought the cost of 5 crore to 30 lakhs or 25 lakhs. But even at 25 lakhs in this country is a lot of money. 25 lakhs, not everybody has, right? And then we all then think how to manage that 25 lakhs. Is it really worth it if my patient relapse after 6 months? How we answer? And there are patients who will relapse. There are patients who will not even get benefit, because only 70% get benefit. If you are in 30% bracket, your money is gone waste. If you have sold the land or sold the jewelry, and now I am in this space, so now I hear those stories, and the patient come and ask, what is my chance? Now, as a scientist, I cannot give them false promise. I have to tell them 70% chance, and even after 70%, there's 50% chance you will relapse after a few months. And then they make a choice whether they want even drug or not. And these are very harsh conversations. And as a scientist or as sitting in the room, it's very easy to sound, but when you face the patient, they sometimes bring, I remember now people see in the news and all, sometimes outside company you see patient sitting with a small kid, give it. And now those are very difficult, but as a scientist, again, I'm just telling you, you have to handle those things. So how to reduce the cost? I think this was the question many people ask us. So question is, we are right now giving it very, very low cost, but cost versus price is a difficult conversation. I think I have less time, I guess. So I will answer these questions if you have the question. I will be happy to explain. But the cost to manufacture is a different aspect. Pricing is a very different altogether. How cost and price are different, we can discuss if we have the conversation. But that dilemma is still there, and I will be happy to take that question. Okay? And the major thing when I take the question, just to give you what drives the CAR-T pricing, many people think this is the cost. That's the only cost. So if you see, many scientists especially, this is for scientists and colleagues, many times you feel if I do this part, I will reduce the cost. That means if you do manufacturing, you may reduce the cost. That is not the answer. Main cost comes from these. Even if you solve this puzzle by a box or by creating a facility in your own institute, that is not going to help you, because maybe you get vector from somewhere, CGMP lab you make in your backyard, and then start doing QC and start sample. This is only one part of the puzzle. What you have to do, product development, whether it's a basic research which you are doing, but IND enabling study, which are phase 1, first in human trial. You have to go through all clinical development pathway. You have to make all the financing cost, wherever the money you get from, right? Then all the overhead cost that comes with the regulatories, corporate, education and outreach, etc. And that is the thing which will put the price different and the manufacturing cost different. I will not go in detail of this. Just to summarize now, what we have done is India's first CAR-T therapy. We reduced the cost. These are unique, which we told you it is comparable efficacy. So efficacy is as good as any other CAR on this planet Earth. But toxicity-wise, we feel with our early data, we are better. Current status is now at least CAR-T is accessible, affordable at certain level, 90% at least cost we have reduced. Of course, there's a long way to go. We have to further reduce the cost in this country if we want to give the CAR-T to many people. Challenges are still remains. Even majority of our patients, despite these efforts, can't afford. We need lot of support from the government, foundations, etc. Even the GST, the life-saving drug costs 12% GST, 4 lakh rupees even in the GST it costs. So how to get rid of those burdens, I think it is all upon all of us, how we work together and make it work. So CAR T-cell therapy at least marks the entry of India in the cell and gene therapy. At least we can proudly say that India has a cell and gene therapy, although just recently, but we have it. It is safe, simple, and scalability is there. So that's the puzzle we have solved. We are providing at scale, right? And we have learned from the different discipline to ensure that CAR T reaches to the market. Right? Those were our inspiration, like Mars mission, or the other examples, even Dr. Talwar's vaccine model, how he worked hard and got those vaccine in the beginning. Right? So what the future lies? Future is, I think, in the combination, in the collaboration. Cancer, we cannot win by one drug. We have to now start studies which are combination therapy. CAR T alone will not work again. Many patients will relapse and it will be then failed model. So we need to combine many modalities, whether it's radiation therapy, small molecules, biologics, checkpoint inhibitors, you name it. And many trials are happening, but a lot more is needed. We need CAR T for solid tumor or different type of malignancies, not just for one CAR T for leukemia and lymphoma. And ultimately, I think we would need allogenic CAR. Whenever it comes, we have to be adaptable, as I said. Many technologies will change in next 10 years. Many new technologies will come. We have to be ready to adapt fast so that we can reduce the cost and move on to the next level. Like CRISPR, for example, even material-based gene delivery molecule. So those are the new frontiers. We have to keep an eye and keep adaptable. And last, all the work is done by the postdocs, PhD student, project staff, as well as scientists at Immunoact, and all the funding agencies, Tata Trust, Vadvani, BIREC, ICMR, DABT, DST, all the people who supported the work, Lawrence Lab, who also invested, and all the wonderful collaborators, whether it's UPenn or NIH or MD Anderson or our own colleague from Tata Hospital, including IIT Bombay. Really thanks to all of them who ensured that this project is successful. And finally, thanks to all of you for listening so patiently. Thank you.
Thank you, Professor Purwar, for the very comprehensive and engaging lecture and sharing your very inspiring journey with us. We have time for just 1 or 2 questions from the audience.
Thank you. So you have said that it has been now mature process, right? So how do you review the technology? You have used the same technology you have used for other diseases, right? And second question is that about that when the cancer has the 2 types of therapy, either you can go with checkpoint therapy or you can go with cell therapy or gene therapy. Which one you prefer?
Yeah. I will answer the second question first and then come to first question because that can be detailed one. So question is that when you have 2 types of therapy, one is checkpoint inhibitors and another is CAR T, which one you will take? So it depends on the cancer type. Checkpoint inhibitor, for example, will not work in the blood malignancies. So then there's no point of discussing checkpoint inhibitor as a first place when they don't even work. So every drug is for a particular indication. Checkpoint doesn't work directly, but of course they can enhance certain immunotherapies effect, but as a alone modality, they don't work. So with CAR T, they can work. Without CAR T, for example, they will not work. So that is a straight answer, right? So here the choice is CAR T. Second question, you said how this technology is different. Or if this is same technologies, then what causes the toxicity is low, right? So if you see that in the world, there are 3 CAR T products, which are again, all of them are against CD19. If you see Novartis product, this has 4-1BB as a co-stimulatory domain. This is considered a different product. Then Gilead product that has CD19 and CD28 has a co-stimulatory domain. So both product considered different because structure of the product is different. Okay. So that's why both have to run those IND enabling studies and go to the market. Third product is again 1941BB, but it is a similar ratio of CD4 and CD8. Right? So that even just by dividing the same ratio, the product is considered different, and all the studies have to be performed, and you know that efficacy and toxicity is again different in that product. Now fourth is us. After them, we have humanized CAR-T. All three are having the scFv that is coming from the murine monoclonal antibody. We have the humanized scFv, and we have 41BB similar to Novartis. So this product is considered different. The construct is different. So once your construct is different, then you have to done entire preclinical studies. So in preclinical, we had shown that our CAR-T produce less interferon gamma, and therefore they induce monocytes to produce less IL-6, right? So if you have less IL-6, that means less CRS and less toxicity. But that was only in the mouse model. So this became a hypothesis, and this is exactly what we have proven in phase 1 and phase 2 clinical trial. When we have treated these now 70 patients, and we have shown that CRS grade 3 is very little, and leukemia patient we see, lymphoma we see is in 0%. Neurotoxicity in both the indication we saw almost 0%. So that's how these are different, and that's the rationale, scientific rationale for lower toxicity.
Yeah, Dr. Purwar, very specific question. So first of all, wonderful, comprehensive presentation, and congratulations to you and your team to reach at this stage and bringing India in the global map. So just I wanted to know that the kind of partnership you had with NCI, was it a partnership, collaboration, or it's about the outsourcing of the facilities or GMP at that point of time, because that was not available with you in the country? So I just wanted to know that what kind of partnership you had with them.
Sure. Thank you so much for the kind words. And so our partnership was always a knowledge-based partnership. So in that partnership, there was no money involved, no outsourcing involved. It was all knowledge-based partnership. It was a scientist exchange program. When we went to NCI, we went for our money. When they visited us, they came with their money. So this was a purely scientist exchange and subject matter expertise discussion. So they have done the clinical trial. They have treated the first patient. They are pioneer in it. So when our clinician wanted to design the clinical trial, we discuss with them, is this clinical trial design is good, or what is their feedback? Similarly, when we stuck with, for example, one patient, even first 2 patients we treated, we were not getting response. And then we were thinking, what is the issue? So when we discussed with them, they said, your flu-cy, which is a conditioning regimen, this is not like proper. You just do 3 days. That little suggestion changed the next course. Similarly, when my something was not working in the lab, so my student went in their GMP lab, stayed there for 7 days. They were very generous in opening the door for us. So there we learned how to manufacture in CGMP environment. Good thing happened with us because of their association. We kept same quality standard as per NIH. So for our regulatory and our acceptance, I think, in the outside world, that what kind of quality assays we are doing, those are not questionable now. So those are the, I think, benefit, and that's how our collaboration worked.
Dr. Basak.
Hey, Rahul, it has been really wonderful to hear you talk. And thanks for sharing your experience as a bio-entrepreneur. And my question is, I'm a bit intrigued with this relapse, as you understand. And has there been any correlation with, let's say, initial cytokine response or the durability of the CAR T cell that you're introducing in the first injections? Has there been any comment that you can make?
Yeah. It's a very good point. Many people have actually published that if you have good cytokine response, and in fact, because of that, if you have a good CRS, so CRS sometimes considered good thing in the CAR T, because CRS is not really a toxicity, but it is because your cells are killing the tumor, and therefore you have more cytokine storm. In our case, we are a little young at this moment. We have not comprehensively studied. This is work we are doing. We only have done good cytokine profile in our phase 1 patient, but we haven't done in all the phase 2 patients. I think this is something we will do, but we couldn't get time, I would say, to focus more on now analyzing all the patients, whom it worked and why it really worked, whom it did not work, why it did not work. So those are the questions, I think. As I said, we had just finished our trial. We were, as a scientist, I personally do not know how to take it to the market. So our focus changed into that. I think now we are a little bit settled. We will go back. Now we will learn from clinical outcome to the lab. Earlier we used to say how lab work goes to the clinic. Now we have to learn from the clinic, go back to the lab and answer the question.
Yeah, I just have a minor clarification. When you inject back the CAR T cell in the patient, do they multiply in vivo?
Yes.
And for how many cycles you have any idea that they will multiply?
Yeah. So yes, answer is yes, they do multiply, and this is the reason why we don't need to give CAR T again and again. So unlike other pharmaceutical drugs, we always have to take them at a certain cycle, either repeat daily, monthly, or half yearly, or whatever. In CAR T, they proliferate in vivo. That means once they are given, they are called one and done.
And you have any idea for how long?
I'm coming. So the patient who was even 2-year survivor, even we found the cells after 2 years. And if you see the profile, sometime it goes up, sometime it goes down. So they'll go and come back. So it's very similar to vaccine research. Some of you who are in vaccine research, you know that if you have an influenza vaccine, and sometime when influenza happens, cells will come up. When the influenza goes down, it will go back. So it's like memory kind of response you have. But you are absolutely right. Another thing is that in not all patients it will happen like that. In some it will happen. Some your cells may not even live for 6 months even. So in those patients where they don't live longer, cancer will come back. So a lot of work need to be done. As I said, this is the first initial data, and yes.
Thank you for a very inspiring story. The question to you is, in the later part of your presentation, you were saying that you had raised money from a whole variety of, agencies, including government, also private investment. So can you just tell us how you convince these people to put in, the money that goes into doing this kind of work?
That is, again, as a scientist, we are not trained for those kind of activities, especially raising funds, etc. But as a first timer, we had to do it. We had no other choice. So I have gone most likely to many established strategic partners in the country, probably all of them, mostly majority of them, if not all, if you technically it may be incorrect, majority of them. But thing was that thing like CAR T, gene therapy, people don't believe. There was no trust that yes, we can do it. And this trust to financial world comes with the success story. If you want to raise fund for any other things, like there are so many good examples of unicorn, right? They are not doing something like extraordinary. They are doing very simple, but investor knows that with this simple stuff, they understand, first of all. Second, they know they will get money out of it. So return on investment is guaranteed, mostly. Right? In this space, there's complete black hole. If they invest something, everything may fail. That risk appetite in Indian investor is at least not there. It is coming. Again, everything is changing for good, but at our place when we were talking, always the question was, is this really will work in the clinic? If you fail, what will happen? All the asset you create, ultimately you fail. So nothing will come out of it. But finally, you try to find 100 people, one you will find who want to help. So Dr. Satya Chava, he invested in us, and he now make a lot of money. Right? Yeah. In fact, second time again, they did not let anybody else come.
Okay. Anyway, congratulations on this very wonderful journey and its exposition. One thing which is really fitting in CAR T cells is that every individual person has to be, you have to make them fresh, so to say, even though you have a platform. So are you planning any progress towards, let's say, common platforms like NK cell based and other things? Are you already on it, or what do you expect on that?
Yeah. Actually, it's a very, very good point, and it's a huge challenge, not even a minor challenge. In our country especially, it's a huge, huge challenge, that how to make this entire circle and then still you are treating one patient in that long. And our number of patients are huge, and this strategy costs you like a lot of money. We are working towards it, but with limited success. So I was telling somebody that GD2 CAR we are making from last 5 years. It did not work. So there are many things that doesn't work, because synthetic biology sometimes also luck, because how you make it, it may work, it may not work, and you can't predict really it will work. Right? So many things we are doing. This is one of the success story which come out. NK cell we are trying, but NK cell, the problem is after some time they start dying. You can make in billions, but after billions they suddenly die. They don't get transduced properly with the lentiviral vector. You can't make NK CAR so effectively than the CAR T. Now people are coming up with different envelope, but so those are the challenges. So, but I think something will work out at some point.
I know there are many questions, but that's the time that we have for today. I thank Professor Purwar and the audience for a very engaging question and answer session. I now request Dr. Mohanty to please join us on stage to felicitate Professor Purwar. Thank you again, Professor Purwar, and thank you, Dr. Mohanty. With that, we come to an end to the Foundation Day lecture. I would like to express my gratitude to our guest of honor, Professor Purwar, for taking the time to join us today. A special thank you to our founding director, Professor Talwar, for making this occasion truly special with his presence. I also want to thank all the dignitaries, NII alumni, scientists, researchers, staff, and students for being part of this celebration. A heartfelt thanks to our mothership, Department of Biotechnology, and the BRIC community for being the wind under our wings. Your unwavering support and collaboration have been invaluable to our journey. And none of this would have been possible without the dedicated efforts of each and every member of the NII family. Please join me in applauding the remarkable contributions of our administrative, technical, project staff, scientists, graduate students, our senior manager, Dr. Vashisht, and our director, Dr. Mohanty. A special shout out to Aishwarya for very generously letting us use her brilliant shot of the brilliant architecture of NII on our Foundation Day posters. And now I invite all of you to join us for the Foundation Day lunch being served in the playground and to partake in the festivities ahead. Thank you again.